Hi there, welcome back to Epidemiology Research Methods. This is Module 4, and we will be going over randomized control trials. In contrast to cohort studies and case control studies, randomized control trials are experimental studies. This means that there is some sort of intervention given to subjects by the investigator. The investigator could expose subjects with a certain condition or disease to, for example, a new drug or surgery. It does not have to be a new drug or surgery, though. It could be an educational intervention or a vaccine or really any sort of intervention as long as the investigator is enacting it upon the subjects. However, not all subjects receive the investigational therapy. The control group receives a current standard therapy or a placebo. The investigator then observes patients to see whether or not their condition improves. Randomized control trials are commonly used in clinical research. They often include an experimental treatment for a disease. And the benefit to patients is that participating in an RCT gives them access to a new treatment that may or may not be better than what is currently available. The benefit to the investigator is that treatments must be tested for efficacy in patients with the disease in order to become FDA approved. So, RCTs are commonly used in clinical research because they are a very, very common study design in drug trials. For example, let's say we have a double-blinded randomized control trial of drug Z in combination with standard of care chemotherapy in acute myeloid leukemia patients. Patients are randomized to receive either drug Z plus standard of care chemotherapy or placebo plus standard of care chemotherapy. At the very least, patients receive what is currently considered to be standard therapy for their condition. This is extremely important ethically, which we will go over later. Double-blinded means neither the patients nor the investigator know if patients are in the experimental group or the control group. And this is so that all patients are treated exactly the same. When selecting subjects, it is important to first obtain their informed consent. Next, subjects should all have relatively comparable prognostics at baseline, meaning they should be on a relatively level playing field. Subjects are randomly assigned a treatment group, so it's not possible to exactly match each case to a control as we can do in a case control study. To solve this problem, all subjects must meet extensive eligibility criteria so that they are all sufficiently matching. Some shortcomings of RCTs involve ways that bias can end up in the mix. One of them involves protocol deviations. So, ideally, administration of treatments and collection of efficacy data should be exactly the same for all patients, and any deviation from the protocol can lead to incomplete or inconsistent data. Next, non-compliance can also be a source of bias. Subjects may be non-compliant in all aspects of participation, and this is particularly a problem in experimental studies because patients have a degree of responsibility in showing their cooperation. Lastly, loss to follow-up can also be a problem. Just as in cohort studies, subjects can become lost to follow-up. Another shortcoming would be the placebo effect, which is interesting. Patients receiving placebo treatment may experience improved outcomes if they believe they are on real treatment. This is a problem when RCTs are blinded, especially. Patients can convince themselves that they are on real treatment and then suddenly begin to feel better. Ethics are important in all trials, but they are especially important in experimental trials like RCTs. In clinical research, the Institutional Review Board, or the Independent Ethics Committee, review research protocols and monitor research practices to ensure ethicality. An example of poor ethics would be the Tuskegee syphilis experiment, which to this day is considered a dark mark on American public health. A group of Black men with syphilis were included in a study of investigators who were aiming to look at the lifetime effects of untreated syphilis. So they wanted to look at untreated syphilis over a long period of time. The problem was that the men in the study did not give sufficient informed consent. Additionally, at some point in the study, penicillin was approved as a treatment for syphilis, and the men continued to be untreated all the while they had no idea what was going on. This is extremely problematic because these patients were denied the standard of care treatment, and they were also not adequately informed of what was happening to them. Despite these shortcomings, RCTs are often considered to be the gold standard of research studies because they tend to provide a reliable comparison between subjects who receive a treatment and those who don't, therefore providing valuable information about the efficacy of a treatment. Efficacy is a measure of association that some epidemiologists' textbooks talk about, but not all of them. I do want to go into it because I think that it's interesting. Efficacy measures whether or not a treatment works. So if we return to our trial of drug Z plus standard of care chemotherapy, then the rate of positive response in patients receiving placebo plus standard of care chemo is A. The rate of positive response in patients receiving drug Z plus standard of care chemo is B. Efficacy would be the rate of positive response in the control group minus the rate of positive response in the treatment group divided by the rate of positive response in the control group. So this is a little bit different than the measures of association we have already talked about. So I'll give you a minute to think about what each of these results may mean. If T equals 0, receiving drug Z in addition to standard of care chemotherapy does not correlate to better outcomes than standard of care chemotherapy alone. This is the null hypothesis. If T is greater than 0, receiving drug Z in addition to standard of care chemotherapy actually correlates to worse outcomes than standard of care chemotherapy alone. If T is less than 0, receiving drug Z in addition to standard of care chemotherapy correlates to better outcomes than standard of care chemotherapy alone. So, let's say that in our drug Z study, 30% of the control arm experienced a positive response, and 50% of the treatment arm experienced a positive response. This means that the efficacy would be the rate of positive response in the control group minus the rate of positive response in the treatment group divided by the rate of positive response in the control group. This is a negative number, which means drug Z combined with standard of care chemotherapy correlates to better outcomes than standard of care chemotherapy and placebo. So, while efficacy is interesting, it's not necessarily the most common measure of association in randomized control trials. If you can think back to relative risk ratios, that is what is generally used. Here we have another 2x2 table with our exposure being drug Z and our outcome being positive response. As a refresher, the cumulative incidence in the exposed would be those who receive drug Z and have a positive response divided by the entire treatment population. So 54 divided by 54 plus 55. Cumulative incidence in the unexposed group would be those who received a positive response in the placebo group divided by the entire placebo group. So 31 divided by 31 plus 70. Relative risk is the cumulative incidence in the exposed group divided by cumulative incidence in the unexposed group. So let's divide those two ratios, and we get 1.614. So what does this mean? Patients who received drug Z were approximately 61% more likely to have a positive response when compared to patients who received placebo. We could also say patients who received drug Z were approximately 1.614 times more likely to have a positive response when compared to patients who received placebo. Let's take a quick quiz to finish off this module. So why might an RCT be double-blinded? An RCT might be double-blinded to ensure that patients on all arms of treatment are evaluated identically. Lastly, how does an investigator ensure that her trial is ethical? An investigator would ensure this by never offering any treatment arm that is less than the standard of care treatment available. So, we have gone over cohort studies, and we have gone over case control studies, and today we talked about randomized control studies. Next module, which is Module 5, or our last module, we will discuss cross-sectional studies and ecological studies. Thank you so much for joining me, and I will speak with you next time.